Right. Hey everyone. So for this video, I want to talk a bit about the lab and really introduce you to what we're doing before we actually meet in the lab. And the main reason for this is because this lab is a bit more detailed and a lot of different steps and techniques. And in previous semesters, I found that, uh, we were a bit uh, strapped for time. And I don't want you to feel rushed or anything. I want you to be able to, uh, you know, perform the lab and learn the steps and techniques and everything without that, feeling of being rushed. So let's get right into it. This is Lab 5, and really, this lab uh, focuses on two techniques, really. There's a few others, but there's two main uh, experimental techniques that you're going to be performing. The first one is called paper chromatography. And the second sounds pretty similar, but it's very different. Here we go. Spectrophotometry. And so I'm actually going to spend less time discussing what these techniques are and what their purposes are, what kind of data they uh, will provide for us. I'll do that in lab, but I want to spend this video uh, focusing on the first really part of the lab, which is preparing solutions for these two techniques, and specifically solutions where we extract pigment molecules. So pigment molecules that appear different colors, mostly green, because we'll be using lettuce, different types of lettuces to extract these pigments. And for these, with these lettuces, I'm going to assign each group a different lettuce. Now, in the past, we've had really 4 to choose from. So, typically we have 5 or 6 groups, so there might be a duplicate, okay? Um, we've used everything from, or really the 4 tend to be spinach, types of chard, like rainbow chard. Please ignore the barking dog. I'm in my apartment. I'm surprised my dog is staying chill. Uh, chard, rainbow chard, uh, kale, although kale tends not to produce the best. Anyways, we'll we'll get into it. So we might avoid the kale, but point is there's going to be duplicates. So each group will be assigned one of these types of lettuce, and uh, so we're essentially in your group. You're going to prepare um, 2 samples, 2 solutions from that assigned lettuce. And your 2 solutions are going to be used by the entire class. So it's kind of like a class effort. So for this little demo and explanation, let's just pretend that the assigned lettuce is spinach for this particular example. So in my group, if I'm assigned spinach, by the end of this extraction process, I will have created with my group 2 spinach extract solutions, one of which will be used by the class for paper chromatography. The other will be used by the class for spectrophotometry. So let's talk about how to do it. All right, so I'm looking at part 1. And so I'm really gonna skip a few steps because it's just collecting materials, such as a 50 milliliter beaker, which is kind of small. It's like a glass beaker about this big, as well as a 15, not 50, but a 15 milliliter tube. Has a few names, but I feel like I tend to call it a conical vial. But it's also referred to sometimes as a Falcon tube. Falcon is the company, but it's a 15 milliliter tube. It's about this big. And so, really, I've maybe mentioned this in the past, but the first thing you really always want to do before you even start any lab, the first thing you always really want to do is label. You want to label your tubes, your Petri dishes, whatever it is you're working with. And so specifically, we're going to label that conical vial or Falcon tube. Looks like this. We have Sharpies in the lab. You want to label it with 2 things. The first, your assigned lettuce, so let's just say spinach in this case. And for this first tube here, we would label it with, as just on the side, spinach. And this is going to be your first sample, so spinach number 1. Okay. The only other thing you really want to make sure you add with your label, so spinach 1, is your group name. You can come up with a name. You can also just use the first initial of all your first names together. But you really want to make sure you have your group name as well. And the reason is because this tube is going to eventually be in a piece of equipment called a centrifuge, but the entire class is going to be using it. So you want to make sure that your own sample is labeled so you know which one is yours when it's with the rest of the group. And I'm going to pause this for a sec. I want to see if I can, it looks a little dark when I step away. So let me play with the lighting for a sec. Okay, the lighting made it worse. There was like a reflection and made it hard to see the board. So hopefully it's bright enough or you can turn up the brightness on your screen. But anyways, so here's what we're going to be doing. You'll collect like the beaker and the tube, the Falcon tube, label it, and then really, so what you're going to want to do is get a balance, an electronic balance to weigh things. Should be on your tables already. If not, it's probably on one of the benches. And a weigh boat, one of those plastic weigh boats. Put the weigh boat on the balance and then zero it. because essentially you're going to get five grams of your assigned lettuce. So if we were spinach, want to get some spinach and measure out 5 grams. But you don't want the weigh boat plastic thing included in your measurements. So you would put that on first, then hit zero, zero it out. So then when you add the lettuce, it'll only include that in your 5 grams of mass. Once you get the 5 grams, the next thing you want to do is take those leaves, use some scissors, which we have in the room. And you want to cut off stems. You want to cut off any brown areas. Because essentially we're extracting, we want to maximize the pigments we get out of it. And those tend not to have as many. So cut off the stems, toss them in the trash, any brown regions. And then you want to cut up your leaf into a lot of small pieces. You can have it in that weigh boat. Next, you're going to put all of those cut up pieces of your spinach, in this case, into a mortar and pestle. And so a mortar and pestle looks like this. So let me share my screen. Okay, so you should be able to see this now. This is a mortar and pestle, so we have those on the bench. So you want to take your cut up leaves, small pieces, add them into there. Then, previously in the steps, it has you fill up a graduated cylinder with 15 mils of sucrose solution, which is a 0.5 molar sucrose solution. You want to pour anywhere from 5 to 10 mils out of that 15 milliliter graduated cylinder into this mortar pestle to help further remove and extract the pigment molecules. And by the way, use this pestle thing to grind up. So that's what you're really doing. You're grinding up the spinach leaves or your leaves cut up further. So you're really trying to mash it up and extract those pigments. And the sucrose, the little bit of sucrose, 5 to 10 mils, slowly adding into there can help with that process. Okay. The next thing you want to do, get rid of this. Okay. Next thing you want to do is with that ground up solution, you want to pour it into the 50 milliliter beaker, which is pretty small, actually. It's a glass beaker. A 50 ml beaker is going to look like this. It's a glass beaker. But very importantly, and I wonder if I can draw this. Well, you're gonna dump what's in the mortar and pestle that all ground up into the beaker, but you want to remove any solid, bulky material. And so you're gonna use a sieve, which is like a filter, or it's also called like a colander if you cook and you drain pasta. But essentially, we have small little sieves in the lab. And so they're like handheld. And what you want to do is pour from the mortar and pestle, or I'm sorry, hold the sieve above. So it's like this. Hold it above your beaker and pour through the sieve from the mortar and pestle into the beaker, so that you're getting the liquid part into the beaker, but not the solid pieces. That'll stay in the sieve. Now, the lab mentions to use a rubber policeman, the fine mesh sieve and a rubber policeman. I don't know why it's called that. But I pulled up a picture. I know it's called rubber. It's a little, essentially, it's like this glass rod with a rubber end. So let me show you a picture of that. Oops. Okay. So that's what it looks like. It's longer. It's just kind of zoomed into the rubber area here, but it's a glass rod with this rubber piece at the end. So that's your rubber policeman. And essentially, you're just going to use that to help scrape out all of the ground up solution from the mortar and pestle into the sieve and ultimately the beaker. Okay. The next thing you want to do is in your beaker, there's measurements on the side. It's 50 milliliter beaker, but really you want around 15 mils, 15 milliliters. And so what you'll actually do is after you filter and pour it through the sieve, you're going to add sucrose solution into the beaker and get it to 15 milliliters. So if I remember correctly, there's going to be maybe a 10 milliliter mark and a 20 milliliter mark. So you just want to get it roughly halfway there to get to 15. And then after that, you're actually going to, excuse me, pour, let me get rid of this part now. Pour the 15 milliliters, I can write that in. You have 15 milliliters of your extracted liquid, which is gonna be green, of course, because we're using lettuce, from your beaker. You're gonna want to pour that into your labeled conical vial. So this is gonna go in here. Okay. Now, if you pour this in, or rather, sorry, back up. So the ultimate volume, the final volume you want is 12 milliliters. So you want to pour this in and you want it to get to 12 milliliters. And the gradations, the measurements on the side of the falcon tube, the conical vial, they're a lot more exact and accurate than the beaker. But really, when you pour this, you want to get to 12. That's the magic number. So 12 milliliters. Now, let's say you pour this and you get to 10 or something or 9, like some of it stuck or whatever, or you were less than 15, a lot less, who knows? You can add in more sucrose to get to that 12 milliliters. So if it's like 10 or 9 or 11, add in more of that 0.5 molar sucrose solution in the lab to get to 12 mils. Okay. Once you're at 12, this thing is going to be, we can move on to part 2. And we can load this into a centrifuge. So let me talk about that. So you have your sample 1 with 12 milliliters. And it's ready to be put into the centrifuge. we have 3 centrifuges in the room. They're typically positioned in the lab room on each of the benches, and or one in the back, 2 over here, something like that. Here's the key with the centrifuges. When you open up the centrifuge, let me see if I can find an image of one that looks like the ones we have. Found one. Okay. So that's what a centrifuge looks like, typical centrifuge. And so it has that lid on the top. Open it up, and you'll see that you're able to load these conical vials with the screw caps on them, very similar to the ones that we use, these Falcon tubes. Here's the key, though. Here's the most important piece of information. Please don't forget this. And it's just so important. I'm sure it'll be on your lab practical at the end of the semester in some way. So when you look into that centrifuge, you'll see 6 openings. It'll look like this. Not the most symmetrical, but the centrifuge is definitely symmetrical. The reason, when you take your conical vial with the 12 milliliters of your extracted pigment solution, you're going to add it, you're going to put it into the centrifuge. Let's say you put it in here. Here's the key. You have to make sure that you balance the centrifuge. What does that mean? To balance the centrifuge, it means if you put your tube here, you would want another conical vial with 12 milliliters of solution of liquid here. Opposite. So that it's balanced. Not here, not here. It's always the opposites, okay? Here's why. The centrifuge, what it does is it spins around really, really fast. We're going to set it to 1,000 revolutions per minute, RPMs. And if it's not balanced, the force that's generated will make a huge sound. The thing will move. I've heard horror stories where centrifuges have broken through brick walls. It is powerful. So it's so important that we balance the centrifuge that don't bother turning it on. Don't turn it on. Call me over. When you're ready with your tube, I'm gonna make sure it's balanced. I trust you guys, but this is introductory class, and of course I just gotta be there to make sure that that doesn't happen. So it'll be a bit of a group effort as well. So, because we have 3 centrifuges, can have 5 groups or so. So if one group is ready to centrifuge, we'll wait for another group to do it as well. And depending on the class and how fast we get through things, sometimes we'll use multiple groups or like 4 groups. Like that, something like that. I would never use, however, 3 groups. Why? As I said, that's not balanced. You would need something here. Now, I could do 3 groups though, but what I would do, or let's say we had, for example, 5 groups, and we used 1 centrifuge and we had 4, and then the 5th group was a little slower, and that's okay. Should have a lot of time for this lab anyway, since we're going to do this beforehand, or see this beforehand. Let's say we had just one group left over. What I could do to balance it is get another conical vial and fill it with the same amount of volume. That's important. Has to be the same volume. 12 milliliters of water. I could put it 12 mils of water in another tube and just load it in on the opposite side. I hope that makes sense. It's got to be balanced with the same volume in each tube. Very, very important. Okay. 10 minutes. So turn the centrifuge on once it's balanced. 10 minutes go by, and then we can take out our samples. Okay. When you remove your sample, it's going to look like this. So after the 10-minute centrifuge, it's going to look like this. What you'll notice, actually, let me draw it first. Okay. So you'll notice that there's this solid mass at the bottom. And we call this the pellet. So the pellet is this solid mass that sticks to the bottom side of the centrifuge. So the heavier particles, cells, end up at the bottom. The liquid that's above the cell pellet has a name as well, and it's called the supernatant. Supernatant. All right. So after you spin your samples and you've got your supernatant pellet, here's what you're going to do. Some room here. You're going to actually transfer the supernatant, so just the liquid. Do your best to just get that liquid on top. And you're going to transfer it to a graduated cylinder. So that looks like this. So you're going to transfer this supernatant to a graduated cylinder. So just the liquid part. It's very important. You want to keep the pellet in this tube, this original tube, and just transfer the liquid supernatant. Okay, so let's call that step 1 here. Transfer supernatant. And you want 12 milliliters in here. And similar concept as before, if you don't have 12, if you have like 9, 8, 10, you want to get the solution to 12 ultimately. 12 mils. And so to get it to 12, this time you're going to add a different type of solution, a salt solution to get to 12. So let's do it here. So this would be like a step 2. And it's a 0.35 molar salt NaCl solution to get it to that final volume of 12 milliliters. Okay. Keep this tube. We're coming back to this in just a moment. That still has this pellet. So this is our sample number 1 tube. So it's like spinach 1. But after you get to 12 mils here, you're actually going to get another conical vial. So go and grab another conical vial. And let's see if I'll have room for this. Okay. So another conical vial here. And again, first thing you always want to do is label. So when you go and get this new conical vial, this will be your second sample, okay? So this would be like spinach number 2. And because it's going to go in a centrifuge soon with the rest of the class, you also want your group name on there. And so what you're going to do then, after you've gotten this 12 mil final volume here with the salt, so I guess you could call this step 3, transfer this solution into that new conical vial. So this would be 3. And because you measured 12 mils, it should be 12 mils. Okay. Now, this is going to go in the centrifuge again. But before I get to that part, let's just put this aside in a test tube rack of, and we'll revisit it in a moment. I want to talk about this. It's very important here. This first sample that, remember, so you transfer just the supernatant, the liquid, so it still has this pellet. Let's take a look at that for a moment. Okay. In fact, I'm going to erase this too, because there's just going to be that pellet at the bottom and a little bit of liquid maybe, but really you just want that pellet. Now, what you want to do now is you want to transfer in a solution or a type of chemical, and it's called ether acetone. And you want to get this to 2 milliliters, so it's not a lot. But you want to add to this pellet, I guess you could call this step 4. And it's called ether acetone. In the lab, I think it's labeled like PET, P-E-T, acetone, or PET, anything PET acetone, you see that, or ether acetone, that's what we're talking about. And you want to get it to, with the pellet, you want to get it to just 2 milliliters, which isn't very much. Okay. That's done. This guy is done. He's ready now. So, okay. Essentially, this sample is ready for the class. This number 1 sample with the 2 mils of the ether acetone. And so, as we'll see, or essentially, sorry, what you want to do is take this solution now, and there's an ice box in the lab on one of the benches that's going to say sample ones, or sample number 1, so, for paper chromatography. So really important, and I'll tell you why. Our sample ones are going to be used for paper chromatography. So sample 1 is used for paper chromatography. Sorry, kind of ran out of room there on this little whiteboard, but well, I'm OCD, so I'm going to scoot it over. Hold on. Let me kind of flip it too. So, just kidding. Let's just keep it here. So, your sample 1 is going to be used for paper chromatography, and you're going to put it in the box. Ran out of room again. You're going to put it in the box on the side that says sample 1, paper chromatography. Okay. As we'll see, because we're not done yet, we're almost done. But this second sample, the color code here, second sample number 2, sample 2, when we're done, sample 1's ready. You're going to put this in the box, okay? The ice box on the side that says sample 1's paper chromatography. Sample 2 is going to be used for the other technique, the spectrophotometry. Okay, why is it so important that we don't mess this up? that you don't put this guy in box number 2. The reason is because ether acetone. We use these little plastic cuvettes, we call them, these little containers called cuvettes, they're plastic. We use them for spectrophotometry. And ether acetone will melt the plastic. It'll melt and the melted plastic gets all over the spec. It's really hard to clean up. It can break it. So it's very important that you put this in the right box for the paper chromatography technique, not for spectrophotometry. Sample 2 that had the salt, the NaCl, will be used for spectrophotometry. So let's pick up there. This guy's ready. Put it in the correct box. We can move on to this guy. Now this tube is ready to be centrifuged. So we're doing 2 centrifuges today. This guy's ready, sample 2, to be put in the centrifuge. And just like before, after you centrifuge, let's draw it here because I don't want to redraw it because I'm lazy. You're gonna end up with a very similar looking tube. It's gonna have the supernatant, but also this pellet at the bottom. So after centrifuging this one, you'll get a similar looking tube. Now, before we actually kept the supernatant, but This time, after centrifuging sample 2, you're actually going to discard, you're going to get rid of the supernatant. So you're just going to discard the supernatant. So you can pour it down the sink. And by the way, before I forget, and I'll remind you in class or during the lab, uh, everything can go down the sink in this lab except, I just had it written, but the ether acetone, anything with the ether acetone, which is really just going to be that tube. That has to go in the waste container, a hazardous waste container, which is going to be similar to the past labs where we had hazardous waste that needed to be properly disposed of, like a little orange container right by the sink. So when you're done using that with the ether acetone, it has to go in that waste container. It can't go down the sink because it's actually flammable. Don't want anything flammable in the pipes. But this supernatant can go down the sink because it's just salt, NaCl, with the ground up solution. So nothing bad in there. So that goes down the sink. And again, just like before, you want to keep the pellet. So the only difference here is you're getting rid of the supernatant, keep the pellet. Then you want to increase the volume, resuspend this pellet after getting rid of the supernatant with NaCl. Get it to, should be around 12 here. 12 milliliters of salt solution. So you want to, after discarding the supernatant, go ahead and add that 0.35 molar NaCl to increase the volume with this new pellet to 12 mils. Lastly, you really want to mix in the pellet with the salt that you add. So by the way, let me number this. So first would be discarding the supernatant. And then second is adding this 0.35 molar NaCl to get it to 12 mils with the new pellet. And that's your sample 2. You're going to take sample 2 and you want to mix the pellet thoroughly with the NaCl. So the best way to do that is with a vortexer. We also have 3 of those throughout on the benches in the lab room. Let me show you what that looks like. I feel like we've used it maybe once before. I have one pulled up. Okay, so looks like that. It's a vortex. And essentially, the Falcon tubes look a lot like that with the screw cap. You just push down onto the vortexer and it'll vibrate really fast and spin and mix your solution thoroughly. And then once you've mixed it up, you vortex this with this NaCl and the pellet. It can then be placed in the proper number 2 ice box and be used by the class. And I think that's it. Yeah, that is it. Excuse me. So the remainder of the lab is doing these 2 techniques, the spectrophotometry and the paper chromatography, with these prepared solutions. And as we'll see, and I'll kind of tell you this as well in person, each group will use 2 prepared pigment solutions. You can certainly use your own that you've made, like your spinach one here, but then you can also use one more from another group, whether it's chard or kale or whatever. So you would use 2 for the paper chromatography and 2 for the spec as well. And I think that's all I got. Hope that made sense. Let me know if any questions come up and I'll see you in the lab.